Good afternoon, everyone, and welcome to the HNRCA Monday Seminar Series. Today is our last seminar of our spring series. Thank you to all who have participated this semester. We've had an increased number of participants this session, so thanks to those who've joined us new this year. We've been so happy to have you with us. I do want to follow up on the video for Dr. Irwin's talk back in April. We are almost done finalizing the video, so it will be posted soon. Dr. Irwin gave an excellent presentation, so I encourage you to check out the video that you can access through the YouTube link through the HNRCA seminar webpage. That is all for housekeeping, so I can introduce today's invited speaker. We are very excited to have Dr. Evan Rosen, Chief in the Division of Endocrinology, Diabetes, and Metabolism at Beth Israel Deaconess Medical Center and Professor of Medicine at Harvard Medical School. Dr. Rosen's lab focuses on epigenomic and transcriptional pathways in metabolic health and disease with a focus on adipocyte biology, including how manipulation of nutritional factors can promote beneficial gene expression patterns in fat cells. Dr. Rosen did his undergraduate training at Cornell University, and I forgot to mention that last week's speaker was also a Cornell alum. So we are on a little bit of a streak here to close out our spring semester. And I'm mentioning this because I am also a graduate from Cornell. So a shout out to our alma mater. We are thrilled to have Dr. Rosen lead our seminar today. He has extensive experience and a very distinguished career with high impact publications, success in securing grant funding over many years, and many awards during his career. So we appreciate your time today, Dr. Rosen. Thank you for being with us. I'll now hand it over to our presenter, Dr. Evan Rosen.
Well, thank you very much, Catherine. I really do appreciate the opportunity to come in and speak to you and to your group. And go Big Red, as they say, for Cornell. So I want to first apologize a little bit for this whimsical title, Adventures in Adipose Tissue Biology. I know you're probably used to something a bit more specific. The reason I chose that title is because I have two stories I'm going to tell you about, one recently published, one unpublished. And what unites them is they're centering on adipose tissue biology, but otherwise there's not a lot else that links them. And I struggled to find a title that encompassed both topics. So you end up with this. This is my conflict of interest disclosure. So I am a physician scientist. I do still take care of patients. This is a patient of mine, Mr. H. When this picture was taken, he's a 59-year-old African-American man with long-standing obesity, and he's had and has basically every complication of obesity that you can imagine: type 2 diabetes with very variable control and complications of his diabetes and sleep apnea, which gave him right-sided heart failure, and fatty liver disease and dyslipidemia. And we gave him a statin. He developed myopathy and hypertension, et cetera. So he's a poster child for all the bad things that can happen when you have too much body weight. Conversely, this is a patient of a colleague of mine. And when this picture was taken, Miss I was 46 years old, and she also had been diagnosed with type 2 diabetes at a relatively young age and was rapidly graduated from oral agents to insulin. And if you're not familiar with prescribing insulin, those are 700 units or more. Those are elephant killing doses of insulin. So she takes she is extremely resistant to insulin. And despite those very high doses, is very poorly controlled and has complications thereof. And when she had a biopsy of her skin, what was found is that she has almost no subcutaneous fat. This is a patient with lipodystrophy. And what she illustrates is the perils of not having enough fat. So too much fat is not good for you. Not enough fat, also not good for you. There really is a sweet spot. And it implies that fat plays a very important role and really needs to be regulated. And that begs the question then, what is it that white adipose tissue does? I should take a moment and say there are other kinds of adipose tissue, brown, beige, thermogenic adipocytes. I'm not going to be talking about those today. Everything we're going to be talking about today is about white adipose tissue, which is the kind of fat that marbles your steaks, that girdles your midsection, that you think about generally as fat. So white adipose tissue classically is known as an energy storage depot. So it stores calories in times of nutritional affluence, and then it releases them in the form of free fatty acids when you need them later, such as when you're fasting or when you're cold exposed. And that is the classical function of fat, and it's a very important one. But we know that there are many other things that fat does. For example, it has important roles in regulating muscle. There is fat around the heart that serves an important role in cardiac function and may also predispose in some cases to cardiac disease, cardiovascular disease. In the skin, there's a very important interaction between hair follicles and fat. There's also regulation of wound healing by fat. There's a very interesting interaction with the lymphatic system that my laboratory is working on right now, and others have noted as well. Bone marrow has a lot of fat in it, and that bone marrow serves as an energy depot for the developing hematopoietic cells and also has other roles in systemic physiology. And so wherever fat is found, which is in many parts of the body, it seems to play an important role outside of its nutritional storage capacity. And this is an ongoing story. You can open up a journal almost any day these days and find a new function for fat that wasn't previously appreciated. Despite all this interest in fat and the fact that it does so many interesting things, when you look at it histologically, it's a very bland looking tissue. So for example, if you, the picture on the left is just an H&E stain and you see the classic chicken wire appearance of these lipid-filled vacuoles inside the fat, and it looks like there's not a lot going on. And in fact, the drawing on the right is from an early histologist, which sort of shows what they thought they were looking at, which is just a big jumble of these lipid-filled cells without much organization or structure. But we now know that this is misleading. Even though that's what fat might look like on first glance, there are a lot of other cell types that are inside the fat. We know that there's a rich vascular system, a lymphatic system, as I mentioned a moment ago. There are nerves, there's every form of immune cell. There are stromal cells that include fibroblasts and pre-adipocytes and other things as well. So there's a lot going on in the fat pad histologically that isn't always apparent because the fat cells are very large and they dominate the field of view. You can quantify this, in fact, using a variety of ways, and we've done this here in a paper that we published a few years ago. Essentially, this is from mice that have a special genetic marker which allowed us to separate the nuclei of adipocytes from the non-adipocytes within the fat pad. It's not important what the genetic trick is for the purposes of this slide, but the key element that I want to show you here is on the left, which it says that about half the cells within the fat pad are adipocytes. This is a lean chow-fed mice, and depending upon the depot, as you see on the right, in the epididymal fat, it's about 50-50 adipocytes and non-adipocytes, and that ratio can vary a bit in brown adipose tissue or in other depots as well. But let's just say, for the sake of argument, that between 40 and 60% of the cells in a fat pad in a lean animal are adipocytes, and the rest are these other things that I just mentioned. In fact, this is dynamic as well. When an animal becomes fat or a human becomes fat, we know that there's an influx of many different kinds of immune cells. And what that changes is the proportion of the cell types that are adipocytes and non-adipocytes. And so you can see here in this image that we generated a couple years ago now, that the percentage of adipocytes goes down from about 40% to about 10 or 20% in obesity, even though if you look histologically, the field of view is completely dominated by these very large adipocytes, which have become hypertrophic. In fact, they represent a minority cell type within the fat pad under those conditions. So in the first story that I'm going to tell you about, I want to talk to you about our efforts to develop an atlas of white adipose tissue, specifically characterizing the cellular composition of human white adipose tissue. We've done this for the mouse as well and published that as well. And I'll only refer to the mouse in this part of the talk in as much as it relates to, so we can compare it to the human. But I'm going to focus my comments primarily on humans at this point. So what we do is we like to take a big chunk of fat, and this is a very big chunk of fat, and it's being minced by my hapless postdoc. Now we use an industrial sausage grinder to do pieces like this, but most of our samples are between 50 and 100 milligrams, and so this is a little bit of an aberration. And then what one can do is you can chop it up and then expose it to a protease like collagenase and centrifuge it at low speed. And what you've done is broken apart the tissue into its component cells. And the cells that have lipid in them, like adipocytes, will float to the top, and those cells that don't have lipid, like everything else, the so-called stromal vascular fraction, which includes the fibroblasts, the endothelial cells, the immune cells, they all pellet at the bottom in this SVF pellet. And then if you want to do single cell RNA-seq, for example, you can load them onto a 10x machine or a drop-seq apparatus, and you can then get them out and sequence them. Unfortunately, however, you cannot look at the adipocytes in this way because they're large and they're fragile, and they will not withstand the forces within the microfluidic devices. Okay, so this is very useful, and we and others have used this to look at the stromal vascular fraction, looking at the immune component, the endothelial component, et cetera. But you can't really learn much about adipocytes in this way. So an alternate approach that we took was you can take this whole tissue and you can freeze it, and then you can douse it and isolate the nuclei. It turns out that the nuclei of the adipocyte they're just as tough as the nuclei of any other cell. So once you've broken apart the cells, broken open the cells, you can harvest the nuclei of the adipocytes or all these other cell types, flow them out and put them into your 10x apparatus and then sequence them. And this will give you single nucleus RNA-seq profiles of all the cells, including the adipocytes. And so this is sort of illustrating what we did here. You can see that we've collected abdominal subcutaneous fat and also visceral omental fat from humans. We did both methods. At first, we were doing the collagenase digestion and putting through a drop-seq apparatus and looking at the stromal vascular fraction only. And as our techniques got more sophisticated and optimized, we were able to do this other thing I mentioned, take the cells, the tissue, freeze it, isolate the nuclei, and look at it that way. And this is our subjects. We ended up with about 138,000 nuclei from 3 men and 10 women across a range of BMI. The x-axis here is BMI. So you see we have people who are lean in the 20 to 30 range, people who are quite obese in the 40 to 50 range, and a few people in between. And then we got about 28,000 cells using the single cell approach illustrated here. Those were all women and all subcutaneous tissue. The people who are split red-blue up top indicates that we got visceral and subcutaneous fat from the same individuals, from those same individuals. So this is what it looks like. If you're not used to looking at these kinds of plots, this is a UMAP plot. We're showing the data from the single nucleus sequencing on the left and the whole cell sequencing on the right. And what this does is it takes all the gene expression events that you can measure. So there's, I don't know, several thousand gene expression events per nucleus or per cell. And then it takes that several thousand dimensional picture and it compresses it into 2 dimensions and then clusters so that you can see the different cell types based on their degree of likeness to one another. And so, and then we just color the clusters and you can see a couple things right away. The first thing is this big orange blob in the middle. Those are the adipocytes, and the adipocytes, as I mentioned, are only really going to be seen using single nucleus approaches because, as I said, the big fragile adipocytes do not survive the whole cell extraction procedure. So we don't see that big orange blob there. You'll also notice this sort of yellow-green blob down here at the left. That's the mesothelium. That forms the lining of the visceral fat. And we would have seen that in the whole cell, but we did not use visceral samples in the whole cell sequencing, and that's why it doesn't show up there. So I draw your attention to it because I think you will have noticed that that big green blob is not present here, and I wanted to explain why that was. That's not because it's impossible to look at it in that methodology. It's because we didn't use the right samples to see that for that method. Okay, so now we're looking we're breaking this down by depot and by body weight. So the subcutaneous fat are these 2 panels on the top. The visceral fat are the 2 panels below, lean people on the left, obese people on the right. For the purposes of this illustration, I've left out that group of people in the middle who can, just to make this, the differences between the body weights much starker. And what we can see here is that there are some important differences between depots, but we see much less of a difference between body weight for the human. So we'll go back here. So for example, you can see the shape of this adipocyte cluster here looks very similar in lean people and obese people, but it's very different than the shape of that orange cluster in the visceral versus the subcutaneous fat. I hope you can appreciate that, for example. Of course, we only see mesothelial cells in visceral samples, but you can also see other differences. Look, for example, at these cells. These are the adipose stromal and progenitor cells, which are the pre-adipocytes and the fibroblasts and all those types of cells in there. And you can see that this ASBC cluster is very similar in lean people and obese people, not identical, but very similar, but a very different shape going from subcutaneous to visceral fat. There are, by way of comparison, very few differences. There are some that change with body weight. There are more macrophages in the heavier people than in the leaner people, and some other differences as well, but those are dwarfed by the differences across depot. Now, I will tell you that in the mouse, it's the complete opposite. In mice, we see huge differences between lean mice and obese mice, but we do not see huge differences between the depots. Some differences, but not huge ones. So that's an important point of distinction between the human and the mouse, we think. Okay, so this is a relatively large study for single cell or single nucleus approaches. We had about 15, 16 people or so. That's a reasonably decent number of people, but it's still 15 or 16 people. It's not hundreds or thousands. And so one of the things that you can do with this data is you can, now that you know the cellular composition of human fat, you can then go back and look at bulk RNA-seq data sets that were collected as part of other people's studies or your own work, and you can deconvolute them because you know what the profiles should look like for the individual cellular components. And so there are algorithms which essentially let you look at a bulk RNA-seq data set and ask the question, what was the composition of that whole tissue when it was harvested, based on what we know now about cellular composition. And this deconvolution, we performed that on samples from the METSIM cohort. This is a group of 330 men from Scandinavia who had subcutaneous white adipose tissue biopsies. BMI is plotted on the x-axis, and we're looking at the prevalence of the different subtypes of cells, of the populations of cells. And we can see, just as in the mouse, the heavier you get, the lower percentage of adipocytes you get, and the more myeloid immune cells, for example. So we're seeing we can sort of get a sanity check here, and we can see that our deconvolution looks like it's providing reasonable data. We'll deconvolute again later in the talk. Now, what we published was a lot of information about the immune cell component, the endothelial or vascular component, and the ASBC component and mesothelial cells as well. But I want to restrict my comments today to the adipocytes, because I just think that's the part that it was really unique about our work, and that is, I think, of most interest to most people. So what one can do is take this orange blob here, all these are the adipocytes, let's say about 30,000 nuclei, and we can pull them out in silico, and we can then recluster them without the other cell types around them. Let those fall apart and ask, how many clusters of those do we see? And the answer is, we saw about seven clusters, and those 7 clusters have more or less unique markers. You can see, for example, that AD1 and AD2 are sort of devoid of very many unique markers. We would call those the sort of baseline generic fat cells. AD1 is a subcutaneous population, AD2 is a visceral population, but ADs 3, 4, 5, 6, and 7 have unique markers which indicate to us that they represent a distinct subpopulation of human adipocytes within these depots. And, there are things that you can do to sort of reassure yourself that this is more or less correct. A couple things you can do computationally, and then a few things that you can do experimentally. One of the things you can do computationally is you can then take your raw data and you can use a completely different clustering algorithm using a different mathematical basis and ask if you get the same clusters. And by and large, we do see that. You can also ask important questions like, do all the clusters, are they all seen in all the people? For example, if this cluster 6, for example, was only seen in one person, that's not a good sign. You would like to know that these are generic across people. And in fact, we do see that. We see all these clusters in all the people. And so that makes us feel more confident that these represent real types of adipocytes. And then there's some experimental things we can do. We can actually take histological sections and stain for markers of these different subpopulations and ask whether we can see them or not. And the answer, when we could find good antibodies, was yes. So you can see here for AD4, 5, 6, and 7, antibody markers for GRIA4, for example, which is the marker of the AD4 population, demonstrate adipocytes which are GRIA4 positive, and they are surrounded by GRIA4 negative adipocytes. PGAP1 is a marker for the AD5 population, and you can see these sorts of PGAP1 positive adipocytes surrounded by PGAP1 negative adipocytes, and so on and so forth. AD6 marker EBF2 is a transcription factor, so you're looking at the nucleus and not the whole cell. AGMO is a cytosolic protein, and you can see it here. AGMO positive cells surrounded by AGMO negative cells. So again, increasing our confidence that are reassuring us that these are real subpopulations that can exist at any given moment in people. What about the heterogeneity vis-a-vis the depot or, for example, body weight? And so again, we see some important differences across depot. Subcutaneous tissue on the left, visceral tissue on the right. And what you can see is that ADs 2 and 6 are primarily visceral subpopulations. AD 1, 3, 4, and 7 are primarily subcutaneous subpopulations, and AD 5 is found in both subpopulations. So that's kind of interesting. Similarly, we can look across body weight, lean people on the left, obese people on the right. And what we can see here is that AD7 changes quite a bit. It's the one that jumped out to us the most. You can see this red dot in the middle, this red blob here. It's much denser in people who are lean than it is in people who are obese. And we'll get back to AD7 in just a moment, which is, I think, one of the more interesting subpopulations. So what do all these different subpopulations do? Well, we don't have any functional data. It's too hard to flow out these cell types. It's not something we've been able to look at. It's a very frustrating technical point for us. So we have to infer. At present, we're trying to infer what they do. And what the data we have is gene expression data. And so what we're looking at here is for all the different subpopulations, what is how much of each of these types of genes do they express? Genes that encode adipokines, or insulin signaling components, or enzymes that promote lipid synthesis or break down lipids, or thermogenic genes, et cetera. And you can see some interesting differences. So for example, AD6 seems to be a more thermogenic population. AD5 seems to be a population which might respond to insulin better. There are lipolytic populations and lipogenic populations. Again, we're not trying to say that these are restricted to one subtype or another, but they may be these activities may be enriched in one subtype or another, again, based on what we're seeing with their gene expression profiling. And we can look also at some of the specific markers or more selective markers. AD3 and 4 contain higher levels of the enzymes that encode lipid synthesis factors. Again, they're found, these genes are found in the other subpopulations as well, but they're enriched in AD3 and 4. Similarly, desaturases and enzymes involved in desaturating fatty acids seem to be most highly expressed in the AD4 population. So if you're interested in desaturated lipids like palmitoleiate, for example, it may be that AD4 is the subpopulation which excretes that secretes those at most high levels. Adaptive thermogenesis genes in AD6, as I said, and striated muscle development genes for reasons that I don't understand in AD7. Another thing that we can do is we can go back and look at GWAS data. So we know, for example, that type 1 diabetes is an immune disorder. And if we now look and say, among all the different cell populations that we found, where are type 1 diabetes genes most likely to be found? And we see that they're in the immune cell component. So that's a nice sanity check for us. It tells us that we can begin to identify what cell types within the fat pad might be housing the genes that are responsible for certain metabolic traits. Waist-hip ratio, we knew was an adipose tissue related phenomenon or trait, but what cell type within the fat is encoding that? Well, it turns out it's almost certainly the adipocytes themselves. BMI is known to be mostly encoded in the CNS, and so we don't see any significant interactions here. And HDL, LDL, and triglycerides meet nominal significance, and in some cases, like in HDL and LDL, nearly meet genome-wide significance for adipocytes as well. Type 2 diabetes, interestingly enough, seems like those GWAS hits really look like they're in the adipocytes. Again, this meets nominal significance for adipocytes, not genome-wide significance. But what we can do is we can look now at the subpopulations. So we can break apart the adipocyte component and ask which of our 7 subpopulations are most likely to be involved in these traits. Waist-hip ratio is found in all 7 subtypes of adipocytes, but interestingly, type 2 diabetes seems that trait seems to be concentrated in that AD7 population. That's the population where genome-wide significance is reached. We think that's really fascinating because if you remember, AD7 is a minority cell population. It makes up, I don't know, 2 to 5% of the adipocytes within a fat pad, mostly in the subcutaneous fat. And we're very interested in how a minority cell type like this could have such an outsized genetic impact on that. This is another experiment that we did. We took completely unrelated people. We isolated their adipocytes and we did bulk RNA-seq and then we deconvoluted it. And we know the HOMA-IR for these people. So we can categorize these people as insulin resistant on the right or insulin sensitive on the left. And what we can see here is that, again, when we deconvolute these bulk samples, the more AD7 you have, the more likely you are to be insulin sensitive. So that's very interesting. We don't really know what that means. We don't know if it means that if you're insulin sensitive, you're more likely to have more AD7, or if you have more AD7, you're more likely to be insulin sensitive. But this is an important relationship that we want to deconvolute further. And again, when we look at the genes that characterize the AD7 cell population, they include some muscle genes, as I said, but also genes like AGMA, which is a bona fide type 2 diabetes GWAS hit, and suggests that there are specialized functions of this minority cell type, and a very, very interesting area for further investigation. Finally, another thing that we can look at are cell-cell interactions. And again, I don't want to overstate this. The data in our hand are gene expression data. So what we're doing here is we're taking genes that encode ligands, like leptin, let's say, or adiponectin, or many, or any cytokine, and genes that encode receptors for those ligands, and then asking what cell types are making the ligand and what cell types have the receptors. And in that way, we're trying to infer crosstalk within the adipose niche. So this is human visceral adipose tissue, lean people on the left, obese people on the right. And you can sort of look by the heat map and it'll tell you the number of those interactions. And one of the things that you can see right off the bat is that, as we know that there's a much more likely to be immune cell adipocyte interactions in obesity, because there's an influx of macrophages, of activated and pro-inflammatory macrophages in obesity. And you can see that here. These boxes in the obese people are lighter. There's a slightly different color than the boxes in this thing. So, you can see that our data supports this idea that in obesity, there's adipocyte interactions that occur with immune cells. But your eye is probably not drawn to that part of the graph. What your eye is probably drawn to is down here, which is a much more impressive set of interactions. And this implies that there are important interactions between pre-adipocytes, adipocytes, and the vascular component. And that they're there at baseline, and then they get much more intense during obesity. And some of this is known. VEGF, for example, is made by adipocytes. It increases in obesity, and it has important effects on the vasculature. So it's not like this is completely new biology, but I would say that there are hundreds, if not thousands, of papers about what's going on up here with the immune component, and a dozen papers perhaps about what's going on down here. And this suggests to us that there's a lot of new biology to be learned about vascular interactions with adipocytes in the fat pad and how that changes in obesity. And so I'm going to summarize this part of the talk here. We've known that there are three major kinds of adipocytes in humans or animals: white, beige, and brown. The beige and the brown are thermogenic, and I haven't talked much about those today. But within now the white component, there appear to be additional subpopulations. We don't know if these represent distinct cell types or if they're cell states that blink on and off under different conditions. We don't know whether these cell subpopulations are anatomically distributed within the depots in unique ways. We're trying to figure all that out now. But I think that what we've begun to get is the beginnings of an atlas to understand what the components of the adipocyte compartment are. And what are we doing now? Well, we're adding more depots to our list. We're adding more cells and more people. We're looking at people under different conditions. We've now just completed sequencing of humans who have, obese humans who've undergone either an RYGB or a vertical sleeve gastrectomy. And we have adipose samples from them at the time of surgery, that is when they're the most obese, 1 month, 6 months, and 12 months following. And we're beginning to see some interesting changes in cellular composition that occur during weight loss. We're also looking at conditions like lymphedema, chronic lymphedema, where there is changes in the adipose compartment, which turn out to be very interesting and important. And we're seeing some very interesting things in that as well. So we're building on our prior work now with more depots, more disease states, more conditions, et cetera, and trying to really build out the adipose atlas for humans. So we're going to switch gears now. We'll take questions on this part at the end, I believe. And I want to focus now on a story about control of lipolysis by the local release of oxytocin. This is Erway Lees, the postdoc who's been leading this. Many of you may know about oxytocin. It's a hormone that is, it's 9 amino acids long, so it's a nonapeptide. It's made in the hypothalamus, specifically in the paraventricular nucleus and the supraoptic nucleus. And then it is sort of transported down these long axons into the posterior pituitary, where it is released into circulation. And it has important roles in things like uterine contractions leading to childbirth, milk letdown for lactation, maybe some roles in wound healing, and then perhaps most famously nowadays, social behavior. It's been called the empathy hormone or the cuddle hormone, because oxytocin seems to be important for social cognition and other psychiatric and neuropsychiatric conditions. And because of this, there's been a lot of interest in pharma in sort of harnessing the power of oxytocin. either through intranasal usage or perhaps IV usage, or people are developing long-acting forms. It might be, some are resistant to stomach acid. It might be oral. So there's a lot of work in pharma on oxytocin analogs, and they're mostly interested in looking at this in areas like autism, schizoaffective disorder, places where empathy, reciprocity, and social cognition are lacking and really need to be enhanced. But it's also been known for a long time that oxytocin has metabolic effects as well. And for one, it's anorexigenic. And so if you give large doses of oxytocin, appetite decreases, and there may be other beneficial metabolic effects. So that's fine, for people who are getting a slug of oxytocin. But what is the role of endogenous oxytocin? That's an interesting and open question. Now, I should say that the role of oxytocin as a lipolytic agent really became clear in the early 1960s. This is a paper published in Nature in 1963 by Burt and his colleagues. This is the whole paper. I've cut it off, the references and a couple of other words, but basically this is the entire data set. There's no supplemental data. This is the whole thing. And what it shows is that in these, like, I believe it's 5 women who were given Pitocin to enhance uterine contractions at childbirth. And all Dr. Burt and his colleagues did was measure free fatty acids floating in the blood of these women a half hour, an hour, 2 hours, and 3 hours after giving them the Pitocin, and saw a sharp increase in the amount of free fatty acids. And based on this and a subsequent paper, which appeared in a different journal a few months later in dogs and in mice or rats, I believe, Burt and his colleagues concluded that giving someone or an animal a slug of oxytocin promotes lipolysis. Well, we had noticed in our data that the oxytocin receptor is highly expressed on adipocytes, and specifically it's expressed on visceral adipocytes, very high levels on visceral adipocytes, less on inguinal or subcutaneous adipocytes, and very little on thermogenic brown adipocytes. Furthermore, it's on the adipocyte itself. There's a little bit of the stromal vascular fraction, but most of the expression at the RNA or the protein level can be seen on the adipocytes themselves. And we just repeated the experiment that Burt and colleagues did 50, 60 years ago now, by showing that if you take mouse adipocytes in a dish or human adipocytes in a dish and you dump oxytocin onto them, you can induce lipolysis, as here measured by glycerol release. You could do the same thing in vivo. You take a mouse and you squirt it full of oxytocin, just give them a big slug of oxytocin, and you can see glycerol increases and free fatty acid increases. And so what we've seen here is that, yes, it is true, giving oxytocin exogenously causes lipolysis. But this then begs several questions. The first is, how does it work? What we know about lipolysis largely is that norepinephrine or epinephrine works through the beta-adrenergic receptor, works through a GS-coupled protein. Adenyl cyclase is activated. This increases cyclic AMP levels in the cell. PKA is activated, and that causes phosphorylation of hormone-sensitive lipase, perilipin-1, and other factors. And this increases the lipolytic cascade. This enhances and really revs up the lipolytic cascade. In humans, there's another pathway working through cyclic GMP and through the AMP receptor, for example. And that cyclic GMP activates PKG, and it works basically through the same phosphorylation sites on HSL and perilipin-1. But what we found was that when we give oxytocin, we do not see any change in cyclic AMP, nor do we see activation of PKA. And if we give PKA or PKG inhibitors, we don't block the actions of oxytocin on lipolysis. And so this pathway that I've just so painstakingly described to you does not appear to be important or the direct way by which oxytocin induces lipolysis. However, it has been known that mitogens and growth factors and other things can also activate lipolysis through ERK. One of the main people working in this area is Andy Greenberg at Tufts. Sheila Collins also has work in this area. So this is not completely new, but it's kind of a bit of a sub-literature. There's sort of this sort of side literature in lipolysis, which says there are other ways to get lipolysis if you have ERK activation. And oxytocin has been shown in, for example, myometrial cells to cause ERK activation. And so we looked in fat, and in fact, oxytocin induces ERK phosphorylation in a timely way and in a robust manner in fat. And if you block MAP kinase or ERK activation in a variety of ways, you can prevent the lipolysis that happens when you add oxytocin. So oxytocin increases lipolysis, and that's blocked by blocking ERK. So we know that ERK is somehow involved. So we've learned a little bit something about that. We're going to get back to that in just a moment. So in order to assess how oxytocin is doing its thing, we took advantage of an adiponectin specific or an adipocyte specific Cree that we generated some years ago, and we knocked out the oxytocin receptor specifically on the surface of adipocytes. So you can see we got a pretty good knockout. Here you can see the immunofluorescence here of oxytocin receptor on the surface of these cells, and it's gone in the knockout. These animals on chow diet appear pretty normal. They have normal fat mass. They have, more or less normal, behave normally. They look normal. On a high-fat diet, they're a little bit more obese, but really not much more obese. Doesn't look like there's a huge difference in their energy homeostasis. But they have a greatly reduced ability to undergo lipolysis in the setting of being fasted, for example, or being put in the cold. Both stimuli for lipolysis that are somehow not working fully. So you can see that there is some increase in lipolysis with fasting or with cold in the absence of the oxytocin receptor, but it is not as robust as you see when you have intact oxytocin signaling. And when we look molecularly at what might be going on in the adipocytes, we saw reduced phosphoperilipin and reduced phosphohormone-sensitive lipase. And what was puzzling was these are the sites on perilipin and HSL that are activated by pKa. But I just told you a minute ago that pKa and cyclic AMP are not involved in oxytocin signaling. So how did this happen? Well, when we thought about it for a bit, what we thought this suggested was that oxytocin, while not itself directly activating pKa and cyclic AMP, might in fact be sensitizing to those factors. So in fact, we wondered if oxytocin was in a way licensing or facilitating or enhancing the ability of beta-adrenergic stimulation to induce lipolysis. And in fact, that is the case. We have mouse adipocytes here in a dish, and we treat them with isoproterenol, which is a pan-beta agonist, and you can see an increase in lipolysis. But if we pre-treat them with some oxytocin, we shift the curve up and to the left, suggesting that we, at any given dose of isoproterenol, we get a more robust response, and our maximal response is even greater. You can see that as well here. Oxytocin is itself a weak inducer of lipolysis. Isoproterenol is a much better inducer of lipolysis, but the two together gives you something synergistic, much better than additive, if you will. And then if you block ERK signaling, as just to go back to that, you can see that you don't really affect the isoproterenol action very much, but you block this ability of oxytocin to synergize, to enhance this ability of beta-adrenergic stimulation. So that's in vitro. It also works in vivo. You can see the same thing. If you give an animal a slug of oxytocin, it's a weak inducer of lipolysis. Isoproterenol is much better. The two together gives you the most robust activation. And we can ask what happens at a loss of function experiment if we pre-treat the animals with atosaban, which is a pharmacological antagonist of the oxytocin receptor. So we're blocking the oxytocin receptors, and then we give just isoproterenol. We're not giving oxytocin, we're just blocking endogenous oxytocin. We see reduced lipolysis. And we can do the same thing genetically by using the animals I just described that are lacking oxytocin receptor on their adipocytes. When you give them a slug of isoproterenol, you get less bang for your buck. That isoproterenol is less able to induce lipolysis if you do not have an intact oxytocin receptor. And so based on that, we said, okay, this is great. Oxytocin is sensitizing the cells for their adrenergic stimulation. And so what we speculated would happen is that when you really need to undergo lipolysis, such as in times of fasting or in cold exposure, we expected that the pituitary would release a lot more oxytocin, sort of like a suckling reflex. There would be this release of oxytocin into the serum. But we didn't see that. You can see here that serum oxytocin does not change in the cold or in the fasted state. It didn't go up as we anticipated. But Irway, my postdoc, was undaunted, and he actually measured oxytocin in the fat pad itself. He ground up the fat pads and looked at local oxytocin in the fat pad. And surprisingly to me, what he found was that there was probably a doubling of oxytocin levels within the fat pad itself, not reflected in the serum, but locally within the fat pad. There was a huge amount of oxytocin that seemed to be being released. And so that was a really interesting observation to us. I should point out this is true in humans as well. This is data that we generated using samples from Sam Klein at WashU. He took men and fasted them overnight and then refed them for 3 hours and did a subcutaneous fat biopsy and a serum sample at the fasted state and in the refed state. And when we looked at those samples, what we found is that, in fact, there was no consistent change in serum oxytocin when you go from fasting to refed. But there was, in fact, a significant difference in the tissue oxytocin level. So refeeding causes a drop, which is another way of saying that fasting causes an increase in tissue oxytocin levels. So this phenomenon seems to be true for mice as well as for people. So where's the oxytocin coming from? Well, I just spent a half hour, a minute ago, talking about the cellular composition of adipose tissue. And so we had all that data for mice and humans. And when we look for oxytocin producing cells, we don't see them at all really in the fat pad. At first, we thought, well, there must be like an oxytocin secreting fibroblast or an oxytocin secreting macrophage, and that's what's doing this. But it's not, does not appear to be the case. And we did additional work because there's some dropout and it's not very sensitive to use single nucleus RNA-seq for this purpose. Irway took a reporter mice and crossed them to an oxytocin-free mouse and flowed out whatever cells would be reporter positive, tomato positive in the fat pad, expecting to see some of these oxytocin-producing cells, and got nothing. So that was disappointing. But when we sat back a minute and thought about it, oxytocin is a neuropeptide. We know it's made by neurons. There are neurons in the fat pad, but the cell bodies, the nuclei of those neurons, are not in the fat pad, and they were not found in our single nucleus approaches. They're found in the paravertebral columns for the sympathetic chains. And so we then asked whether sympathetic neurons in the fat could be the source of the oxytocin. And in fact, that does appear to be the case. And we have an enormous amount of data that corroborates this, and I'll show you some of it right now. So for example, if we take oxytocin-Cre and to a reporter, you can see these GFP positive neurons. They co-stain with tyrosine hydroxylase, indicating that these are sympathetic neurons. And the merger suggests that these are the same neurons that are expressing both oxytocin and tyrosine hydroxylase. For those of you who are not familiar with how innervation of the fat pad works, sympathetic innervation, there are these, there's the brain, and then there's these, the vertebral column, and then there's these paravertebral ganglia that are the sympathetic chain that send off these neurons that go to the various organs, including the fat. The relevant levels in mice for adipose tissue are T13, L1, L2, which innervate the inguinal fat, the subcutaneous fat, and the aortico-renal ganglia, one of these splanchnic ganglia that innervates the visceral fat. And so Irway went and took mice that had oxytocin-Cre, crossed to a reporter, isolated the aortico-renal ganglion, and you can see here these red dots, which indicate that there are cells that are expressing oxytocin-Cre in the fat pad. And when we look at them, we see that they co-stain with tyrosine hydroxylase, telling us that these truly are sympathetic neurons. We could do the same thing in the sympathetic ganglia that innervate the inguinal fat. For example, at L1, you can see these GFP, it's a different reporter here, so you can see these GFP positive dots here. They're red here and cherry positive. We don't see them in ganglia that do not innervate fat, and they're also tyrosine hydroxylase positive. Now, there's two problems with the data I just showed you. The first is that in those animals, the oxytocin-Cre is on the entire lifetime of the animal. So it's possible that the Cre activated the reporter sometime during embryogenesis, and it's not on anymore. It doesn't mean that oxytocin is being made at the time when we do the experiment in the adult animal. So that's one caveat. The other thing is that those ganglia innervate lots of organs, not just fat. So how do I know that those red neurons are going to the fat? So this experiment that I'm about to show you answers both of these questions. What we take is an oxytocin-Cre mouse, and we inject directly into the fat pad an AAV2 that will travel up the axon, go to the cell body, not cross the synapse. And if Cre is active at that moment and present, will cause a rearrangement which allows the M-cherry cassette to be expressed. And so we see the M-cherry positive cells in these ganglia, and what that tells us is that Cre has to be on at the time we did the injection at the adult mouse. So the oxytocin allele gene is on at that moment. And it also means because we did the injection in the fat pad itself, that those neurons go to the fat pad. So we answer both these questions with the same experiment. And we did the same thing in the inguinal fat, and we got the same result. There are other things we've done as well. You can artificially set up a genetic system that will allow you, using a chemical trick, to activate the sympathetic nervous system. And we've done that here. And when we force the activation of the sympathetic nervous system without, for example, putting the animal in the cold, but just by adding this drug, then what we see is, glycerol goes up. We've activated lipolysis. We do not see a change in serum oxytocin, but we do see a change in the tissue levels of oxytocin. So we see more oxytocin in the EWAT and in the IWAT. And then finally, the last thing that we did was we took the oxytocin gene, we floxed it, and then we knocked out oxytocin, specifically in tyrosine hydroxylase neurons. We did a lot of work to show that we're not affecting oxytocin expression in the brain and the hypothalamus. That's all intact. These animals basically have normal food intake. They have normal body weight on chow and high-fat diet. And when we look at them, we see that their serum oxytocin is unchanged, but they now have reduced oxytocin in the fat pad. And when we fast them or put them in the cold, actually put them in the cold first or fast them, we see reduced lipolysis. And similarly, if you give them a slug of isoproterenol, they are unable to fully respond to that isoproterenol because they lack oxytocin in these peripheral neurons. And so I would summarize what we found in this very simple diagram, that there is a subpopulation of neurons, of sympathetic neurons, that derive from the parasympathetic chain, the sympathetic chain that co-express norepinephrine and oxytocin. We've done some careful quantification. I haven't shown you that data, but it's about between 9 and 13% of the neurons, sympathetic neurons within the ganglia are co-express oxytocin and norepinephrine. The norepinephrine, of course, works through the beta-adrenergic receptor, cyclic AMP, PKA. This is the classic pathway. Oxytocin working through a different G protein coupled receptor, oxytocin receptor, activating ERK. And then there's a bit of a black box here. We don't know yet how the ERK and the PKA are interacting with one another. Andy has some prior work to show ways that this might occur through a sort of hierarchical phosphorylation approach. And we're interested in looking at this further. This causes lipolysis. And so that's sort of our model, is that oxytocin and norepinephrine are cooperating to allow lipolysis to happen to its full extent. So that's the data I wanted to talk to you about. The people who did the work, I mentioned Irway, who really has spearheaded this oxytocin project. Margot Emont is a fellow in the lab who really spearheaded the single cell work. Of course, the single cell and single nucleus work involves a lot of people, including patients and the surgeons who give us a sample. But we have a technical staff that enables us to collect the samples and process them, et cetera, and bioinformaticians who are excellent at doing the bioinformatics, et cetera. Linus Tsai is my partner in crime in this. Our collaborators at WashU, at Rockefeller U, and at MGH, and of course our funders as well. And with that, I will thank you, and I hope we have, I believe we have a few minutes left to answer whatever questions you might have.
Thank you, Dr. Rosen, for that wonderful presentation. Yes, we have time for questions. So a reminder to attendees, you can use the raised hand feature in Zoom. We will unmute you, or you can submit your questions by typing them into the Q&A box. Let's start with the Q&A box. First question is from Kelly Cara. Have you looked at adipocyte subtype profiles in people who previously had obesity but are now lean? If not, what profile might you expect to see in this population? I'm curious if their subtype profiles, in the lean state more closely resemble profiles in the obese state, which might give us some clues to weight gain fluctuations or cycles in these individuals. Yeah, no, it's an important point. So we have now collected and performed the sequencing on patients who are obese and looked at 1 month, 6 months, and 12 months after their either Roux-en-Y or their vertical sleeve gastrectomy. And the data are being analyzed, and I don't have a lot to tell you about that, but we will be able to tell you what the cellular composition is of and the adipocyte subtypes of these people as they've gone from the obese state to the lean state and their metabolism improves. So I don't have much to tell you about people, but we have a comparable experiment in mouse that is much farther along, and we're about to write the paper now, and I can give you some information from that. So obese mice who had either vertical sleeve gastrectomy, performed by Randy Seeley at University of Michigan, or who were given semaglutide. Okay, so these mice become leaner and metabolically more healthy, and we've looked there. And what we see in the mouse is that they have, obese mice have a certain subpopulation of adipocytes, couple subpopulations, which are express markers of hypoxia and fibrosis and inflammation, and those seem to revert. And there are important changes that we're also seeing in the immune component and in the pre-adipocyte component as well. So we're starting to see changes in these mouse models. What's interesting in the mouse is that we see some changes that are changing with the reduced body weight, and other changes that are occurring either with VSG or with semaglutide, but not with both.
So there are modality dependent and independent changes that we're observing. And I think we'll see similar things in humans as well. So stay tuned, a lot more coming on that.
All right. Our next question is from Jennifer Lee. She sent a second comment. I don't know if she's negating the first question or not. I'm just going to read it. Hi, Evan. Excellent talk. Thank you. Considering the technical challenges that prevent functional outcomes in adipocytes, have you or others done follow-up studies to see if transcriptome signatures change and if the composition or proportion of subpopulations shift in the same patients following weight loss or gain?
Yeah, so this just back to the same question that I just
Okay.
answered. So the answer is we'll see. So we'll see what happens with weight loss. With weight gain, that's not a study we've done in humans where we've made them fatter, but we've looked at, as I showed, lean people and obese people, and we can see differences between them. We see more, as I mentioned, we see more important or more larger differences between depot than we do between lean and obese in human. But that's what we see.
Okay. I'm going to go to Andy Greenberg, who has two questions. I'm going to start with the first one, though, just so we can make sure to try to answer everyone's questions. Hi, Evan. Extremely elegant study. Previously, we demonstrated that TNF treatment of human adipocytes activated ERK, which resulted in downregulation of phosphodiesterase 3, which led to increased CAMP levels and phosphorylation of PLIN1 and HSL. Is that a possibility here?
So could the oxytocin effect be working through reductions in PDE3B? I guess in principle it could, but you would expect that to be manifested by increased cyclic AMP levels in the cell and increased pKa, and we don't see that. So it seems to me unlikely, Andy, but I guess it's formally possible. Why TNF should do that through ERK and oxytocin might activate ERK and not do that is I'm not sure, but that's commonly seen in the ERK field, that different inducers of ERK activity can have different outcomes.
Okay, I think we have enough time. I'm actually going to just continue with Andy's second question.
Sure.
A few years ago, Phil Shear published that inflammation produced in adipocytes is healthy. Do you have insights in lean and obese adipocytes about adipocyte production of cytokines with your Atlas studies?
Yeah, it's an interesting question. Clearly, there is an increase in cytokine production in the obese adipocyte, but there are some cytokines that are present in lean adipocytes as well. I would say that, again, we don't have functional data from these people. We just have their gene expression profile. So it's hard I don't want to get too far out on a limb and speculate, but so I'll just leave it at that. And I'll just say we do see interesting cytokine profiles there. And then changes, of course, in the immune cell component so that the fat pad itself is awash in a different group of cytokines, different clusters of cytokines in the lean state and in the obese state. So I can't really connect what Phil's done yet, but our data is consistent with some of his ideas.
Great. Okay, I did see a raised hand from Alice Lichtenstein. If you still have but it was up for a while. So if you still have a question, can you just raise your hand again and I'll get back to it. The next question is Larissa Constantino, who asks, does stimuli that trigger OXT secretion lead to a clinically significant lipolysis?
The stimuli, so the stimuli you might be referring to would be uterine contraction or suckling, I guess is what you're asking. So there is, in fact, some data that suggests that during suckling there's an increase in lipolysis, and in fact, if I'm going to go a little bit out on a limb, I think that this is why the system is like this. For example, is that why should oxytocin be involved in lipolysis? And I think it probably has to do with the fact that oxytocin is causing milk letdown. And then after milk is let down, you have to synthesize new milk in the epithelial cells of the mammary gland. And that requires that you, liberate fat so that you can make milk, make milk fat. Milk fat, some of it is produced endogenously within the epithelial cells, but quite a lot of the fat in milk comes from the adipocytes. It requires lipolysis. And so I think this is teleologically makes a lot of sense then that oxytocin would be a driver of lipolysis. under these conditions. And I can go further and say we have a preliminary observation that this is correct. If we look at mother mice, dams, who are oxytocin receptor null on their adipocytes, their pups are runted relative to pups from wild-type mothers. And so that suggests that those pups might be getting skim milk. And so we're doing some experiments now with Bridget Gregg at the University of Michigan, where she's milking our mice. We're doing sort of lipid analysis and nutritional component analysis of the milk. And I suspect that the animals that don't have oxytocin receptors on their adipocytes will have low-fat milk, but we'll see.
Hey, great, thank you. Our next question is from Shangdong Wang, or it's two-part question. One, can age or sex affect white adipose 7 cell types or their expression pattern? And 2, can mice lacking OXT receptor develop less non-alcoholic fatty liver if you feed mice with a high-fat diet?
All right, let's do the first one first. So we don't really have, we have a range of ages from 20s to 70 or so in our human samples. But there are so few people in each age range that it's really hard to draw conclusions about age. So I just don't want to go on a limb there. And our mouse studies were all done at the same, their mice were all the same age. So I don't have much to say about age. Sex, I can say something about. Originally, what we did was we did human women and mice, male mice. So when we saw differences between the human data and the mouse data, we didn't know whether it was a species difference or a sex difference. So we went back and we did male humans and female mice and did all the stuff again. And basically what we can conclude is that by and large, the species differences outweigh the sex differences. That is not to say that there are not some interesting sex differences. And we're looking into this more now when collaborating with Karen Rui and others to do sex specific sequencing and trying to understand the basis of that. So I would say sex has a smallish effect. Age, I have no data on. The second question was about oxytocin. Can you repeat the second part of the question?
Yes, mice lacking an oxytocin receptor develop less non-alcoholic fatty liver if you feed with high-fat diet.
Yeah, so right, so what you're driving at is that lipolysis is adipose tissue lipolysis is required to provide lipid that is used by the liver to make fatty liver. So is getting rid of lipolysis in the fat through ablating oxytocin action, would that be a strategy for reducing fatty liver disease? And the answer there is I really don't have the data. The animals that lack oxytocin or lack oxytocin receptor are a little bit fatter on high-fat diet, but not a lot fatter. And we just haven't looked yet at those livers, but it'd be very interesting to do so. Because we're thinking along the same lines that you are, but we just haven't done it yet.
All right, I think we have time for one or two. Robin Wilson says, very interesting work. Does increased oxytocin have any thermogenic effect?
Yeah, really interesting question. And I can answer that pretty definitively at this point. We've looked at this a lot. And what I can tell you is that the effect on lipolysis drives a thermogenic effect, but there's no independent effect on thermogenesis. So in other words, if you put an animal in the cold and the animal does not have access to food, then it will, an oxytocin receptor null animal, if you disrupt the system, they will get cold very fast because they can't liberate free fatty acids to burn from their white fat. So it looks like they have a thermogenic effect. But if you feed the animals when you put them in the cold, now you ablate that effect completely, right? So in fact, and we've done more work on brown fat, there are very few oxytocin receptors on brown fat cells or beige fat cells, we know that. And in general, I would say oxytocin does not have a direct effect on thermogenesis, but does have an indirect effect on thermogenesis via lipolysis, by providing the substrate to burn during cold exposure. So that's my somewhat tortured and complicated answer to that question.
Thank you so much. This has been great with questions. We have time for one more, so this will be our last question. Richard Alpert says, did you observe any differences in response to OXT in male versus female adipose samples?
Yeah, good question. Everything I showed you was in males. So all the data presented was male animals, but we have gone back and done most of the all the important experiments in females. So the knockout of oxytocin in the tyrosine hydroxylase cells, the knockout of the receptor on the adipocytes. We've gone back and done female cohorts, and we get the same data. So it's essentially the same. So also not much change in body weight, but there are big changes in lipolysis, and they appear to be roughly equal in size between the males and the females. So I don't think there's a major sex effect there.
All right, fantastic. So that was our last question for today. Thank you so much, Dr. Rosen, for answering those questions. And that will conclude today's seminar. So thank you to the audience for attending, and we'll see you next time.
Thanks very much.
Bye.
Bye.